The central dogma of molecular biology, as the name implies, is the established principle that consists of underlying processes involved in protein synthesis, and that illustrates the flow of genetic information. This involves replication, transcription, and translation. Replication is the first step in central dogma that involves creating two copies of parent DNA in semi-conservative, semi-discontinuous, and bidirectional manner. Later it will be better explained and illustrated. Assuming that this is our parent eukaryotic DNA, and this is our legend wherein the white represents deoxyribose, red for phosphate, orange for adenine, yellow for thymine, green for guanine, and blue for cytosine. Primarily, this is how the DNA appears: double helix. The replication will not start if the DNA is on this configuration. Therefore, it needs to be untwisted and unzipped, that is, being catalyzed by the enzyme called DNA helicase. Another enzyme involved in the initiation stage of replication is what we call topoisomerase, that relieves torsion strain in the untwisted parts or strands and prevents supercoiling. Finally, the untwisted DNA. Eventually, this will be unzipped by DNA helicase. Single-strand binding protein, or in eukaryotes, RPA replication protein A will bind in each strand to prevent reannulate. So as you can see, this shows you a unidirectional manner. This is just to represent one side of replication, although in a real case scenario, it should be bidirectional. In each strand, primase will synthesize RNA primers that will provide starting points for the DNA polymerase. This will be followed by the synthesis of oligonucleotides in each strand through the action of polymerase alpha in 3' to 5' direction. However, we have a 5' to 3' strand. Therefore, polymerase alpha will move backwards, creating a lagging strand due to RNA-DNA oligonucleotides called Okazaki fragments. This makes replication semi-discontinuous. The spaces between Okazaki fragments are called nicks, which are being filled by the DNA ligase with nucleotides. As the polymerase alpha moves and synthesizes nucleotides, polymerases delta and epsilon are responsible for proofreading and correcting the base pairing. As the process proceeds, RNA primers will be removed by flap endonuclease 1 or FEN1 and will be replaced by the corresponding nucleotides by the action of polymerase alpha. Eukaryotic replication terminates at the telomeres of chromosomes through the action of telomerase, and that's how the replication flows. Transcription is a process by which the RNA is being synthesized from the DNA template strand. This occurs in the nucleus of eukaryotes, similar with replication. The initiation of this process involves the detection of TATA bases from the DNA template strand or Tata box, after which Tata box binding protein or TBP along with transcription factors or TFs will bind on that particular region. Eventually, RNA polymerase will bind beside the Tata box. Before we proceed, these are color representations for transcription. Assuming that the aim of this transcription is to produce a messenger RNA, furthermore, assuming also that the Tata box has been bound with TBP and transcription factors, and the RNA polymerase II is bound beside it. The 3' to 5' DNA template strand separates from the non-template strand as it passes on the RNA polymerase II. As the deoxyribonucleotide of the template strand passes through the active site of the RNA polymerase II, the RNA polymerase II synthesizes the corresponding ribonucleotide. Simultaneously, the RNA polymerase II performs proofreading. As the encoding for the pre-mRNA proceeds, the binding of elongation factors takes place in RNA polymerase II, causing the continuous 3' to 5' movement of RNA polymerase II. As the RNA polymerase II approaches the end of the template, CPSF or cleavage and polyadenylation specificity factor, CSTF or cleavage stimulation factor, and other release factors bind to the RNA polymerase II, causing the release or detachment of pre-mRNA from RNA polymerase II. Finally, this is now our pre-mRNA, which will undergo post-transcriptional modification. This involves 3' polyadenylation and 5' methylguanylation that will protect the mRNA and facilitate its transport towards the cytoplasm. Moreover, introns will be spliced out and exons will be retained.